All right, welcome to the final video for Chapter 3, where we are going to finish up with our discussion on organic molecules, and we're going to finish with nucleic acids. So we've talked about an overview, carbohydrates, lipids, proteins, and now the final organic molecule group, nucleic acids. Nucleic acids, like proteins and carbohydrates, are made up of monomers that eventually form polymers, so your longer chains of the organic molecule. For nucleic acids, we have two different types of nucleic acids. One is DNA, which is deoxyribonucleic acid, and the second is RNA, ribonucleic acid. And these nucleic acids differ in the sugar molecules that are part of their general structure. And these nucleic acids are the genetic material in organisms that are that is giving the information of what proteins need to be made, how many of those proteins need to be made, and the kind of expression of those proteins in our bodies. And so proteins kind of run those cellular processes, govern those cellular processes, and DNA, nucleic acids, and RNA tell us how much of those proteins to make, or tell our bodies how much to make. In nucleic acids, that DNA resides in all of the cells of the living organisms. And DNA is made up of these long fibers called chromosomes. And on every chromosome, there are multiple genes that make up a specific segment of the DNA that's a part of that chromosome. And the DNA has the information that tells our bodies what amino acid sequence should be part of that polypeptide. So here we have a DNA molecule, which is part of a chromosome, and that one segment of the DNA molecule is going to make up the gene. The gene will be transcribed from DNA into RNA, and then that RNA will be translated into a protein. And we'll talk about transcription and translation in a future chapter. But for each of the DNA and RNA molecules, those are the polymers of the nucleic acids, and those polymers are made up of monomers called nucleotides. And each nucleotide has three specific components that make up that specific molecule. Each nucleotide is made up of a 5-carbon sugar, and that 5-carbon sugar determines whether or not it's DNA or RNA. So DNA is going to have the deoxyribose sugar, and RNA is going to have the ribose sugar. The sugars will also have a phosphate group attached to that molecule and a nitrogen-containing base, so a nitrogenous base. The nitrogenous base determines, the nitrogen-containing molecule will determine what nitrogenous base that the nucleotide actually is. And so the nucleotides for DNA can either be adenine, guanine, cytosine, or thymine. And those are referenced by A's, G's, C's, and the letter T's. And so, for example, this nucleotide shown here on the slide is a thymine nitrogenous, a thymine nucleotide. It has the thymine nitrogenous base, it has a deoxyribose sugar, and then it has a phosphate group. Okay, and these single nucleotides can be put together to make a complete nucleic acid, DNA or RNA molecule. the way these nucleotides attach to one another is this phosphate group will attach to an adjacent sugar molecule on an adjacent nucleotide. And then those nitrogenous bases, again, determine what type of nucleotide is in that segment. And so, like we mentioned, each DNA nucleotide has one of four possible nitrogenous bases. It could be adenine, guanine, thymine, or cytosine. And this is for DNA specifically. And so if we look at each of those bases individually, you can see that adenine and guanine have double ring structures. Thymine and cytosine have single ring structures. And here, there are also pairing rules in DNA that are really specific. So a double ring structure will always pair with a single ring structure. And in this instance, for DNA, adenine always pairs with thymine, and guanine always pairs with cytosine. Okay, and for nucleic acids, you have dehydration reactions that are going to put these nucleotides together, and it's the same dehydration reactions that we've talked about for carbohydrates, lipids, and proteins. These are specifically linking the monomers of the nucleic acids together and making those polymers. And the monomers, remember, are the nucleotides, and the polymers are the nucleic acids, so those polynucleotides. And again, these monomers are put together through bonds that form between the phosphate groups of one nucleotide and the sugar molecule of an adjacent nucleotide. And this ends up forming what we call a sugar phosphate backbone that makes up the kind of structural component, the rigidity of the DNA molecules and the RNA molecules. And the nitrogenous bases actually kind of like stick out or hang off of the sugar phosphate backbone. And this allows them to interact with the other DNA backbone that makes up the double helix, the double helical structure of the DNA molecule. And DNA molecules are double-stranded. They have two polynucleotide strands, and these strands coil around one another to form this double helix structure. The bases along the DNA strand form or are held together through hydrogen bonds. Okay, so like I was saying, the nitrogenous bases within the double-stranded molecule of the DNA are held together via hydrogen bonds, and we'll show that exactly how that looks in the figure on the next slide. And remember, I said that each of these nitrogenous bases are going to specifically pair up with one another according to specific pairing rules that allows us to always determine what base pair the nucleotide would be paired with if you only have one strand of DNA. And that those base rules are, again, adenine's only pairing with thymine, and guanine is only pairing with cytosine. Okay, and so if you have a DNA strand sequence that is GAATGC, what would be the sequence of the other strand? So you can pause the video, kind of go through this, and I'll give you the answer here in a second. Okay, so hopefully you were able to figure it out. With this specific sequence, the sequence for the other strand would be C, T, T, A, C, and then G. And that's because G always pairs with C and A always pairs with T. This is going to always be the case for DNA. RNA differs a little bit in that RNA does not have thymine. It has uracil, which is a U. So in that case, for RNA, A always pairs with U, and G always pairs with C. Okay. All right. So here in this figure, we have an example of a sugar phosphate backbone of a DNA strand. You have a single nucleotide, another single nucleotide, and those are going to pair with the second strand according to those pairing rules. So the A's are paired with the T's, the G's are paired with the C's, and you can see those hydrogen bonds forming between those different nucleotides. And that's what holds those two strands together to form that double helix DNA molecule. And so like we mentioned, DNA and RNA are polymers of nucleotides. Both are made from those single nucleotide monomers, and those single nucleotide monomers always have the same similar structure. So a phosphate molecule, a sugar molecule, and then a nitrogenous base. For DNA, those nitrogenous bases were adenine, guanine, cytosine, and thymine. For RNA, those nitrogenous bases are adenine, guanine, cytosine, and uracil. So the thymine in DNA is replaced with uracil in RNA, and then also the sugar molecule is different in RNA in that it's not a deoxyribose, but it is a ribose sugar. So those are two big differences. A third difference is that DNA is always a double helix structure unless it's being replicated, and then only then are certain parts split and then put back together. So they're very rarely single strands. And the RNA is always found as a single strand, whereas DNA, again, is usually that double helix structure. And so here we have the nucleotide structure for RNA, where you have your sugar, which is now ribose instead of deoxyribose, same phosphate group, and your nitrogenous base. The difference here is a U instead of a T. And then we'll talk about, whenever we go over the DNA chapter, how DNA is converted into RNA, and then how RNA is translated into amino acids to make those proteins. And so that's it for our RNA video and discussion on, not RNA, but nucleotide video. So our nitrogenous bases are our nucleic acid organic molecules. There we go. Maybe I'll learn how to talk in a little bit. And so that's it for the video on these organic molecules. And this is the last video for our organic molecule chapter. So chapter 3 in the Simon book, and chapter 2, section 3 for the OpenStax free book that's available online. So let me know if you have any questions. You can send me an email or write your questions in the discussion section on CAT courses. And so that's it for this week's information. And we will see you next week when we talk about cells and the structure of cells.